this is a really valuable paper, kind of laying out the hallmarks of aging. These are some of the basic principles that contribute to the biology of aging. And it won't be a surprise to anybody that this is where we focus most of our energy. One, because the mitochondria are just really cool, right? They're this really cool self-contained organelle. We like to think of, they control most things going on in the cell. But from a more practical perspective, really they sit either at the center or connected to all of these other hallmarks. So by better understanding what's happening in the mitochondria with age, and then actually intervening to target the mitochondria, we feel like we can affect a lot of these other hallmarks. So another part of what our lab does is study skeletal muscle aging, or sarcopenia. So that's the loss of muscle mass and function with age. So when you think of aging or an older person, this decline in muscle tone, muscle function, muscle mass is kind of one of the first things we think about. It's one of the most identifiable aging phenotypes. And this loss of the ability to generate force and power is a real contributor to this increased risk of falls, as well as this exercise intolerance and frailty, which then is associated with comorbidities and just generally poor quality of life. And lastly, for this slide, one of the main points, we see a lot of fat infiltration and a lot of metabolic defects that are associated with skeletal muscle aging. And this is true regardless of activity level. So this person on the left, Ed Whitlock, was a marathoner. He was running marathons into his late 80s, right? Super healthy guy. He's clearly got aged muscle, right? So although physical activity is critical to staying healthy, muscle ages regardless. And you can see this in several studies now have published data similar to these, where they look across, let's see if I can coordinate this here. They look at cross-section of ages here, and then they look at thigh area. I'm not going to do this. You see this decline with age going, in both muscle area on the right, on the left, and muscle strength on the right. And these slides, they showed a gradual decline in both. I prefer to think of this as there's a plateau up until about the mid-50s before you really start to decline. And as I'm, in my mid-50s, I'm feeling more and more strongly about that. But regardless, this clearly shows that there's a clear decline in both muscle mass and function, and the function kind of declines a little faster than the area. And this is a multifactorial condition. A lot of things feed into this decline in muscle function with age. But where our interest in mitochondria and muscle converge is that we really think that the mitochondria kind of play a role in all. Anybody that has been to more than 2 aging talks has seen this slide. And I just announced to my lab yesterday, this is the first time that I have ever shown this slide in a talk. Almost all of these things cause sarcopenia. And the mitochondria play a role in all of these things. And so the way we like to think of mitochondria in the lab, it's either a cause or a consequence of most things that happen with age, right? So it may not be the primary driver, but it's an amplifier of the pathology, right? Yeah, I wouldn't say that I wouldn't say there's one cause. There are nodes where you can intervene, and I think I'll talk about what one of those nodes might be today. Yes. But this interchange between mitochondria and muscle and aging is borne out by several large studies. And this is from the SOMA study relating the mitochondrial energetics in skeletal muscle with cardiorespiratory fitness and mobility. This other one is relating it to walking speed. And there's more and more coming out from these large cohort studies where they're actually looking at the relationship between skeletal muscle energetics and other factors of health. So in this case, they're looking at cognitive impairment and biomarkers of Alzheimer's disease and relating that to muscle function. Again, this is a large cohort study where they're looking at relationships, right? So there's not a cause and effect arrow here, but there's a clear relationship. And And then this is a study from, I think, 20 years ago, where they looked at people that had really high metabolic fitness, which is these dark gray bars, and then low metabolic fitness, these light gray bars, and looked at the risk of death across these 6 chronic conditions, and see that going from high fitness to low fitness almost doubles your the risk of death across the span. So with all this in mind, what we're trying to do is really ask the question, or can we target the mitochondria to improve muscle function and general health? So the idea is we're trying to see, can we come up with interventions to go from here to here, getting out of this disability threshold? So the questions, I think I've kind of beaten this to death. Are the mitochondria a promising target for improving muscle function? And then, can we use mitochondrially targeted interventions to learn more about the basic biology of aging? So it's not just a one-way arrow, but can kind of circle back from the clinical trial back into the mice. So in order to pursue these questions, we were fortunate enough about maybe 10 years or so ago to start working with a compound called Elamipretide. A lot of you guys have probably heard about it. Several people here work on it. Mariah talked about it about a month and a half ago in this very room. But it's this tetrapeptide that was discovered accidentally that associates with the mitochondria by interacting with this fatty acid called cardiolipin on the inner mitochondrial membrane. This is a really important component of the mitochondria because it helps to structure the cristae membrane, or the inner mitochondrial membrane, while the business part of the mitochondria works. And these cardiolipin molecules also interact with all of the complexes of the electron transport system and help to sort of arrange these protein assembly into supercomplexes to make more efficient mitochondrial function, as well as altering the properties of the membrane. And this is some work that's really just come out in the last couple of years, which is really neat. Our first experiment we ever did with Elamipretide, came out of a big collaborative meeting, and it was almost like, okay, well, if this is how it works, then this should happen, right? So Mike took young and old animals, gave them an injection of Elamipretide, and then measured their in vivo mitochondrial function an hour later. And so what he found was, when he looked at the young and the old, there was a significant decline in the mitochondrial efficiency. So the P to O went down, indicating there was greater proton leak. The mitochondria were leakier. And after an hour of treatment with Elamipretide, this went back up to young level. Same thing when we looked at maximum ATP production. This went down significantly with age, back up after an hour of treatment. So these are really rapid changes that he was observing. And then later, Matt Campbell, who's now an instructor in our lab, did a similar study to sort of build on this, and then he did an 8-week treatment and basically saw the same thing. Reduced P to O ratio with age, restored after 8 weeks of Elamipretide treatment. Same thing with ATP max. Then Matt went on to look at skeletal muscle function. So here we're looking at the tibialis anterior. And in this case, the muscle is exposed, we tie off the tendon, and then we can stimulate the muscle directly. So this keeps the circulatory and the nervous input intact. And then he looked at animals, older animals that were either treated with a control or Elamipretide in blue, and saw that the Elamipretide treated animals were more fatigue resistant. So associated with the improved energetics, we saw that the muscle had greater endurance capacity. And this translated to treadmill running time. So these are measures at baseline in the old animals and the young animals. And then after 8 weeks of treatment, there was no effect in the young animals, but in the old animals, they ran significantly longer. And in spite of these differences up here, we didn't see a change in the maximum force that the muscle could produce. But more recently, Gavin Farrow, who's a postdoc in the lab, did a study where he looked at the gastrocnemius muscle, stimulated the muscles in vivo, and measured force over this force frequency range. So this shows sort of early, low levels of muscle function. this is where a lot of your sort of activities of daily living happen in here. Very seldom do we function at maximum force production. So what he saw in the old animals, there was a significant decline in this maximum force. But in the old animals treated with elamipretide, where he really saw the difference was in the submax force production here. So in spite of not seeing a change in the max, in sort of this functional range, there was still an improvement in muscle force. And these next two slides just show that these effects sort of are broader than muscle. They also expand to the cardiac and kidney, which you saw about a month ago. So these are some data by Ann Chow when she was when she was a postdoc in Peter Rabinovich's lab, where she looked at aging mouse cardiac function using echocardiography and saw that the old animals had significant diastolic dysfunction here, shown by this low E to A ratio. And then after 8 weeks of treatment, this was significantly improved towards the young level here, shown in orange. And then similar for the myocardial performance index, which is kind of you just think of this as an integrated measure, incorporating a couple different parameters of function. And here, 8 weeks of treatment brought this back to a lower value, which is more towards the young. In the last year, Gavin Farrow also sort of expanded our ability to do echocardiography using this global longitudinal strain analysis and looked at systolic function. So this systolic strain is a measure of ventricle deformation during the systole. And this goes down with age and after treatment is restored by SS31. There we go. Yeah. And this next slide is you've seen before, most likely. But essentially, the take home is that Mariah showed back when she was a postdoc, I believe, that animals treated for 8 weeks also had less kidney injury. So this was reducing the kidney pathology associated with aging. So this is a measure of glom injury. 8 weeks later, this goes up. It doesn't change in the animals treated with elamipretide. And these are all things that our lab is currently focusing on right now. So I kind of went through this first part relatively quickly. It's all published data, and probably a lot of you have seen some of the data before. But basically, to summarize, that these age-related mitochondrial energy deficits can be reversed with elamipretide treatment. And this is associated with improved function kind of across multiple physiological systems. And what was really striking to us was that these rapid improvements in energetics are similar to what we saw after 8 weeks, which really kind of shakes up how we think about an intervention might work. Most things that target the mitochondria target something upstream and either induce mitophagy or induce biogenesis and require protein synthesis and activation of pathways, where whatever's happening here seems to be happening relatively quickly. So taking this idea that we're seeing these rapid changes after quite a bit of lobbying from me, I worked on STEL for a while to get them to try to support a study where we capitalize on this acute effect. So finally, they supported a study, it was a phase 2 randomized controlled clinical trial of elamipretide in older adults. So what we did, and the study was led by Kevin Conley. So we screened people for average to low mitochondrial function using the criteria of an ATP max. So this is maximum ATP production below 0.7 millimole per second. You can see this is the population from several clinical trials. You can see that this 0.7 kind of differentiates right down the middle from the high functioning and the low functioning. Then we gave these people a single dose of elamipretide and measured in vivo energetics again and muscle fatigue resistance. Immediately after the infusion of the drug and then 1 week later. Oh, and this is just the flow diagram. So again, we used a phosphorus NMR to measure the energetics, and we were looking at the hand muscle here, which is this muscle that causes your finger to move like this. And see it in, there we go. And I'm happy to answer questions about why we studied that muscle if you want after. But then we looked at mitochondrial energetics right after infusion. And this is a plot of the change in the ATP production post-infusion compared to baseline. And we saw an increase within a couple hours of the infusion. And then after 7 days, this went back down to baseline level. So again, it's getting in there, doing something fast, rapidly doing it, and then at least in terms of the mitochondrial ATP production, then it's washing out. But we also looked at fatigue resistance. And this is our homemade instrument for measuring muscle force and fatigue. And so the assay here was just asking subjects to repeatedly do voluntary contractions at 70% NVC as long as they could keep it up. And then once they couldn't do it anymore, that was the end of study shown here. And our measure of fatigue in this case was we just counted the number of contractions they could maintain. And so interesting, what we found was there was no effect immediately after infusion, but when we looked a week later, muscle contraction continued to improve and fatigue resistance continued to improve. So to summarize this clinical side of things, basically the take home is that these rapid improvements in energetics that we saw in the mice translated to older individuals. So the next question is a question that, we were wondering, but every time I gave this talk for quite a while, people asked the question, so we know that Elamipretide interacts with this membrane, but does it actually sort of do sort of a more typical drug targeting where it actually interacts with specific proteins, right? So in order to address this question, we started a collaboration with Jim Bruce. Jim Bruce and Juan in Jim's lab in the UW Genome Sciences. And they've developed a really cool approach. It's called cross-linking mass spectroscopy, where they can actually look at how proteins interact with each other, and in this case, with small molecules. So in this case, we used the biotin labeled SS31. And SS31 is another name for Elamipretide. And then took advantage of the presence of this lysine residue in the normal structure. So we took the hearts from old mice, isolated the mitochondria, incubated them with this biotinylated SS31, then incubated them in the presence of these, oh my gosh, I'm going to forget the name, protein interaction reporter molecules that Jim's lab has come up with. And these molecules form cross-links with lysine residues. So we capitalize on the fact that there's a lysine in SS31, and there are lysines in proteins. And so with this reaction, any molecule of SS31 in close proximity to a lysine in a protein is going to get cross-linked with this reporter. Then you do a sort of a typical protein digestion, and then do an enrichment by pulling down with the biotin tag. And then what you're left is this complex with your biotinylated SS31 and your peptide. Then you do your mass spec and your normal analysis, and then your structural analysis, and identify what proteins interact with this drug. So what we found was there were 12 specific mitochondrial proteins that bind with the drug. And these are associated with oxidative phosphorylation and ATP production, or alpha-ketoglutarate processes. And what was interesting, all of these proteins have been reported in literature to associate with cardiolipid. And this is kind of where they fall in the cristae membrane. So these are the complex 2 sites, complex 4, the cyclooxidase, the ATP synthase, a couple matrix proteins. But we, for multiple reasons, were particularly interested in this ADT1, which it's also known as the adenine nucleotide transporter. So this protein is responsible for bringing ADP into the matrix of the mitochondria and then pumping ATP out to the cytosol where it can be used. And what's really cool about this approach and this mass spec that they do is that not only can you identify the peptides that interact with, the proteins that interact with the peptide, but you can actually identify the sites. And so you know where the peptide is in the protein. So what they found is SS31 interacted with lysine 272 and lysine 33, this peptide. And then Juan did some modeling to identify the most likely position of the peptide in this protein, and basically found that it sits in this channel. So as I mentioned, the adenine nucleotide transporter is a transporter, brings ADP in, takes ATP out. But also one of its functions or side effects is it's also a main proton leak channel in the mitochondria, because this channel can open up and be permeable to protons, especially in the presence of fatty acids. And so based on the modeling, we were hypothesizing that SS31 can come in here and alter the structure of this transport channel to inhibit the proton leak. Then also, well, the next question I guess we want to ask is, does it also affect the ability to transport ADP? And the modeling studies and the structural studies are ongoing about that. But we wanted to look at it, go back to the energetics, right? Back to where I'm more comfortable. So we went back and looked at some of our energetics data. So these are reanalysis of our in vivo spectroscopy. And rather than looking at the maximum ATP production, we wanted to look at the sensitivity to ADP. So you can think of this as the ability of the mitochondria to respond to energy demand in the cell. And so reanalyzing these data, we saw that, so in the gray is the young, the black is the old. So we see a shift of this curve indicating that the mitochondria are becoming less sensitive. But then in the old treated with elamipretide, we see this opposite shift indicating a significant increase in the sensitivity. And that's just quantified over here. Now, the more rigorous scientists out there are going to say, hey, wait a second, you're drawing a line through a couple points here. I don't really believe that very much. So recently then, again, Gavin Farrow, there we went back and looked at this in permeabilized muscle fibers and isolated mitochondria. So in this case, he isolated muscle from these 26 and 28-month-old wild-type mice, either treated the tissue during the permeabilization or during the mitochondrial isolation with elamipretide, or treated the animals for 8 weeks and then did the permeabilization or the isolation. And again, found that compared to the wild type, the elamipretide treatment significantly increased the sensitivity of these mitochondria to ADP. So that is, as ADP increases this way, their rate of respiration increased more quickly in the presence of elamipretide. So this would be consistent with better transport of ADP from the cytosol into the matrix where it can be used. But because this respiration is a complex process that involves the electron transport system, ATP synthase, and the ANT, Gavin wanted to know specifically whether this was elamipretide was affecting the ability of ANT to actually transport ADP. So here he teamed up with Ian Sweet to actually measure the import of radio-labeled ADP into the matrix of the mitochondria here. And then could also do this in the presence of a specific ANT inhibitor. And so what he found when he looked at both in skeletal muscle and heart mitochondria from old animals, that in this range of this low ADP range, this is a sort of moderate energy demand of skeletal muscle range of ADP, there was a significant improvement in the ability of the ANT to pump ADP into the matrix of the mitochondria, suggesting that this is a main driving force behind this increased sensitivity of the mitochondria to respond to energy demands. And these data suggesting that ANT, or showing that ANT actually is transporting ADP better, are also consistent with work done by Huilang Zhang, when he was a postdoc with Peter Rabinovich, where he looked at cardiomyocytes isolated from old and young animals here, and then incubated the old cardiomyocytes in the presence of elamipretide and measured their respiration in the seahorse. And what he found was that they were going from the young to the old, there was a significant increase in proton leak, similar to what we saw in vivo back in the beginning of the talk in vivo. But then when they were treated with this peptide for, I think these were an hour or two treatment, really short-term treatment, this reduced this proton leak. And also when he used ANT-specific inhibitors, this also prevented the proton leak, indicating that this is happening through the ANT. And interestingly, they did not see any difference in maximum respiration here. Again, supporting a role that, maybe a lot of the energetics improvements that we're seeing with this treatment are due to changing the sensitivity of the mitochondria to energy demand. And so to conclude here, basically, going back to take a closer look at our energetic stuff, and then this cross-linking mass spec really suggests that elamipretide is interacting with specific mitochondrial proteins, and that this direct binding of elamipretide to the ANT may be an important part of its mechanism of action by increasing sensitivity to ADP and reducing proton.